Hey everyone, welcome to week 3 of physiological psychology. This week we're going to focus on nervous system cells. In this first video, we are going to talk about neurons, and then we're going to talk about glial cells and other things associated with nervous system cells in subsequent videos this week. In last week, week 2, we really talked about the nervous system in general, the different branches, and then the anatomy of the brain and other parts of the nervous system. So in this week, we're kind of almost taking a step back to look at the building blocks of the cells that create these structures. So let's start with neurons. Neurons are specialized cells in the nervous system that receive, conduct, and transmit electrochemical signals. And those are really big words. It'll make more sense later when we really start talking about the different types of chemicals that are associated with neurons and really what they're doing, like what their function is. And for the purposes of this video, we're really more so talking about the history and the structure of neurons. We've got motor neurons, and for motor neurons, the cell body or soma is actually in the spinal cord. And so those neurons are conducting impulses to muscles for movement mainly. And then we've got sensory neurons that are sensitive to particular types of stimulation, whether that be light, sound, touch. If you think about the different senses, sensory neurons are associated with those senses. We have about 86 billion neurons in the adult human brain. Just think about that number for a second. This image from our text sort of highlights the breakdown of this 86 billion. So we've got 16 billion in the cerebral cortex alone, 69 billion in the cerebellum, and then the rest of the brain has less than 1 billion. And then our spinal cord also has 1 billion neurons. So that's a lot of neurons, right? So just think about that just all these neurons in your nervous system. Let's move on to the history of neurons. How do we even know that neurons exist? Well, Santiago Ramon y Cajal, he won the Nobel Prize in 1906. And what he did is use a staining technique that had been discovered using silver salts to stain infant brain cells. And he chose infant brain cells because infant brain cells are, of course, smaller. So he used those cells so that they could fit more easily on a single slide and that he could observe what was going on with those underneath the microscope. He examined these cells and documented his findings. And even to this day, his images or sketches are still used. I just did a quick internet search to see if I could find any of his original drawings. And this is an example of one that I found online. And as you can see, it's pretty close to what we see in our textbook. So his sketches are still pretty spot on from what we see today in diagrams of neurons. So now let's move on to neuron types, or really, I guess we could refer to this as different terminology you may hear associated with neurons and how they work. So the term afferent is referring to information being brought into a structure, and efferent is basically the opposite. It carries information away from a structure. So the textbook gives us an example that every sensory neuron is efferent to the nervous system, meaning that it's bringing information to the nervous system. So if we experience a sensation, that neuron is bringing that experience of sensation to the nervous system, which again, our central nervous system is our spinal cord and our brain. The efferent neurons is basically the opposite. So every motor neuron is efferent to the nervous system. So if our brain says, hey, move your hand, that's our nervous system, our brain, our central nervous system telling that part of our body to move. So it's efferent to the nervous system. One other bullet point here is intrinsic or interneuron is another term you may see, or type of neuron. You can think of it, whatever clicks for you best. Is when somebody uses that term, they're talking about a neuron that is entirely within one structure. So the textbook gives the example of the hypothalamus. If a neuron and all of its different pieces, which again, we're going to go into the structure of the neuron in just a moment, but all the different pieces that we're going to talk about, dendrites, axons, all these different parts of the neuron, if they're all within that one piece of the brain, that one structure of the brain, then it's considered intrinsic to that structure. I went ahead and put this image in here to help further explain efferent and afferent. So this is an example of an efferent neuron in relation to this neuron, which would be afferent. And the textbook is a neat little trick that you can use to sort of remember the difference between these two types. Is you can remember E meaning exit, which would be efferent. So information is exiting that neuron to someplace else, whether it be another neuron or somewhere else. And A for admitting information or admit. So that would be the afferent, which in this diagram would be this information receiving or admitting information into that neuron. All right, so cell structure. It's helpful before we really delve into the actual structure of the neuron to talk about just general cellular structure. So as many of you know, one of the recommended prerequisites for this course is just general biology, because it is related to the different cells that we're going to see in the nervous system to have a general basic knowledge of cell structure in general. So most animal cells have a nucleus, which contain our DNA. Mitochondria, those are the different pieces in the cell that are helping with metabolic functions, providing energy for the cell. Ribosomes, which synthesize new protein molecules. And then the endoplasmic reticulum, which help transport newly synthesized proteins. So let's take a look at that. I've actually highlighted the relevant concepts that are in our text that we just went through in bullet point format. I highlighted those in blue in this picture here. And all of the other information, it's good information, but it's beyond the scope of this course. So this is really from your textbook the only things you need to know. We've got the mitochondria again, which again that's related to metabolic functioning of the cell. Here's our nucleus. That's kind of that center of the cell that contains our DNA. And then I'm not going to ask you to know the difference between rough and smooth endoplasmic reticulum, but in general, just knowing that endoplasmic reticulum is related to transporting newly made proteins by ribosomes. And if you are interested, the rough endoplasmic reticulum, it's rough because it's got ribosomes attached to it, and the smooth doesn't have those ribosomes attached. Okay, so we've talked about some basic cell structure. Now let's go on to the neuron structure itself. To me, the neuron is just such a beautiful cell. It's beautiful, it's tree-like, and it's got a lot of different pieces. So I'm going to blow up this picture in just a moment, but let's just go over the definitions of the different parts of the neuron. So we've got dendrites, which are receiving information, or the signal, which again, some of these terms will make more sense as we get further into what neurons do and how they function. But the dendrites are basically, you can think of as the receptors. They are receiving information from other neurons. Soma, or cell body, which we kind of went through in just basic cell structure, that has most of the metabolic work going on there. So that's the cell body. The myelin sheath is an insulating fatty layer that will help speed transition of different signals from neuron to neuron. And so within that one neuron, the myelin sheath is going to help the signal go faster. Axons are the conducting fiber that convey an impulse, and they can actually be greater than 1 meter long, which is pretty neat. And if you think about how long the spine is and how long an axon of a neuron would have to be to get that signal up to your brain, it makes sense to think about how long an axon could be. Schwann cells, which help make myelin, and those are actually a type of glial cell, which we're going to go more into later, but I wanted to mention it here because the diagram we're about to look at references Schwann cells. And then axon terminals, those are the end points of the axon. So the axon itself is one point, and then at the end it has different branches which can be considered the axon terminals, or you might hear it be called axon buttons. And those signal the release of transmitters or chemicals that help activate the next neuron, or whatever the next function may be in the process. And then we have the node of Ranvier, which is the space between the myelin sheath on the axon. So let's blow this picture up because I think this will make a lot more sense when you see the picture. So this is a neuron. Again, to me, this is just a really beautiful cell. I love looking at neurons. And so we've got the cell body here, or the soma, whichever term you prefer to use. This is the nucleus. We've got dendrites here. So you can kind of see it's like these little branches that come out, and they are just ready to receive information. Okay, so the information comes in through these dendrites, through the cell body, and then this entire line here, if we could even just put, you know, like some arrows to say, hey, this whole thing right here is the axon. So this whole long stretch is the axon itself. And again, an axon can be even more than a meter long, depending on where it needs to get to. So if you've got an axon from a neuron that's located, let's say like in the sciatic part of your spine, you know, the lower part of your spine, think of just how far that axon has to reach to be able to get that information up. And so on the axon itself, we have the myelin sheath. So that's indicated here in yellow. So remember, it's that fatty insulating layer that's helping electrical signals go through the axon quicker. And the nodes of Ranvier are the little spaces in between. So if you think about the electrical signal that will be coming through this neuron, it kind of bounces. And those nodes of Ranvier are sort of, you know, the space in between where the myelin sheath has been created. And the Schwann cells, which are indicated in that yellowish, or excuse me, that orange-ish color right there, that's an example of a type of cell that is helping to create that myelin sheath that helps that information pass through that axon quicker. And again, there's only one axon in a neuron. However, that axon can branch at the end, as you can see here, if you can see my cursor, that axon branches into what we call axon terminals. And then at the bottom of the axon, some people refer to these as axon buttons. You know, it's basically where the information will be leaving and then subsequently entering either more dendrites for another neuron, or wherever that information is going to be going, or whatever it's supposed to be doing. Which again, there's a lot of different functions for neurons, which we're going to cover in subsequent videos. So that's it for this video. I just wanted to cover the basic overall structure of the neuron. We're going to talk more about functioning later, and we're also going to talk about other cells within the nervous system, and other terminology that is associated with it in this week's reading, which was chapter 1. If you guys have any questions, feel free to let me know, and I hope you guys have a wonderful week. Thank you.